Recording is on.
Okay, is it visible now?
Yes.
Okay. Then we'll start the last part of this module, and well, as Federico mentioned already, the topics will be ANOVA, contrast analysis as a sort of alternative to ANOVA. And finally, something that I'm quite enthusiastic about is a topic of multi-level modeling. Right, there we go. So if you are planning to do ANOVA, the reason why you want to do that is probably you want to know if a treatment, a specific condition, has an effect on an outcome. And most people seem to do that via the frequentist approach, which is what is called NHST, null hypothesis significance testing. And then you apply a t-test and p-values, sometimes f-test, and you have analysis of variance. And what I wanted to show you that there are alternatives that you could consider as well. So if you want to know if a treatment has an effect, then you would like to know, first of all, is there an effect? But secondly, perhaps also how large is that effect, and is it also of importance? So not to be ruled exclusively by p-values, but also by scientific insight. So the frequentist approach, once again, gives all the focus to the data. Yeah? It's about the probability of the data in view of the hypothesis that you have. And that's always, in the frequentist world, a null hypothesis, which states there is no effect, against an alternative hypothesis, which states there is an effect. In the Bayesian approach, we are much more concerned about the probabilities of a certain hypothesis given the data. Which hypothesis is more likely, and you get complete results in probability distributions rather than a point estimate. Then ANOVA is frequently applied analysis of variance, and I don't know if you ever realized that, but ANOVA is actually only a special case of linear regression, with the difference that the independent variables are categorical, like one treatment, second treatment, or a third treatment. So they are not continuous variables, but they are categorical. And what ANOVA then does is it analyzes the differences between the group means in each category, and it compares the variances between and within groups. Now, the following example I took from this website, which I thought explains it very nicely, so you can look it up again if you want to. But what basically is shown here is one treatment A and another treatment B, and we look at some outcome. The outcome is not specified, it's just a simple example. So you see various outcomes for one treatment and another various outcomes for another treatment. And the question is now, are they different? What is usually done in ANOVA is to calculate the group means, what you see here as these blue squares. These are the means of each treatment. And the question is now, are they different? And the first thing you could do is to draw a line, because with 2 points you can always draw a line. And then the next step is you can do linear regression with this result, which means that you make the mean of group A, you make that the intercept, and then you calculate the slope, how much it takes to go from group A to group B. So that's why it is actually just linear regression, the only difference being that you now have categorical variables here. So once again, it is a frequentist approach, so the null hypothesis is there is no effect. So that means that if the slope of the line is about 0, means that the null hypothesis is confirmed. But if the slope of the line is different from 0, is it then the null hypothesis or is it the alternative hypothesis? And to decide upon that, one uses p-values. And once again, the p-values always refer to the probability that the null hypothesis is true. Now, if you do this linear regression for these two groups, you find the intercept, which is the mean of group A, and you find the mean of group B, which is negative because it is lower than the first one. And then we can calculate the probabilities for the null hypothesis. And now we are specifically interested in the slope. And we see that these probabilities are very low, which means that it's not so likely that the null hypothesis is true. In other words, there is a significant effect according to the frequentist approach. So there's another way you can do that. You can also use the so-called F-test. The F-test compares sums of squares, and it compares the group sums of squares with the residual sums of squares. And yesterday we explained what this means, what sums of squares are and what residual sums of squares are. So this is a simple calculation how to calculate an F value. And again, you can then test the ratio between the sums of squares and the residual sums of squares, and that is this value in this particular example, which again, looking at p-value says, well, the p-value refers to the null hypothesis, there is no effect, and you see that the likelihood that there is no effect is very, very low. In other words, it is concluded that there is an effect. In other words, it is a significant effect. So this is how ANOVA works. If you have more than two groups, then the question is, can you still do that? And the answer is yes. But the first approach, just looking at the regression, is telling you that there is a very low p-value. It's even rounded to 0 here, which means that there is an effect. And of course, there is an effect because these are the same groups as we saw before, but we now have a new group here. But it does not tell you which a group has a significant difference. So you can get a better view of that if you look at the estimates of the linear regression, and then you find that the estimate for the group C is very low, and that the p-value is quite high, meaning that here the null hypothesis is probably true. There is no effect, so there is no difference between group A and C, which is also apparent, of course, from the if you look at it graphically. But there are other ways that maybe you have heard of also before. That is a so-called post hoc test. In this case, the Tukey test is used a lot. And then you can make the pairwise comparisons. So you can then calculate specifically for the difference between each group. So in the previous slide, it was an analysis for the whole group, but now you can also compare it with B and A, C and A, and C and B. And then again, of course, you see the same differences. The difference between C and A is not significant. So this is the classical way of how you can study the differences between treatments. That's all, well, reasonably simple. And the only thing I think is maybe new to you, that it is actually just linear regression behind the screens. But there is an alternative, which is especially useful in the Bayesian analysis, and that is called contrast analysis. And this contrast analysis is well, I picked it up from especially the social science literature, and it is, I don't see it so much, or maybe not at all, in the natural sciences. And what is a contrast? Well, it compares, like in ANOVA, one set of means with another to see if there are differences. You can do a t-test, but there's also other ways, and I like to show you these other ways because I think that is quite insightful. So I use two simple examples, one again on apples and another from a PhD student here in Wageningen who studied the effect of hardness of food on the eating rate in humans. Okay, let's start with the apple. The research question that I had was, how is apple weight influencing the size, the diameter, D? So a very simple research question, is there an effect of weight on diameter? Well, if you look at an apple, you can approximate an apple as a sort of a sphere, should be a sphere here. So you can calculate a volume and a density and a diameter from that. And if you do some algebraic calculations, you find out that there is a relation between the diameter and the weight according to a power law. Okay, so here you see that relation. So I have taken 7 cultivars of apples and I have just plotted everything against, so every diameter of every apple against weight. And as you can see, there's indeed this curved relationship as predicted by this power law. And you can also do a regression again, as we saw yesterday, and you can get then these point estimates. But the question that I actually had was, these apples came from different orchards. And the question I had was, is this relation between diameter and weight perhaps different for each orchard? So that's also, you can say, a sort of treatment effect. And the reasons why it could be different is that there are different soils, there are maybe different climates if the orchards are not close to each other, and there's also different management. So the question is this site, the orchard site S, does that have an effect on the diameter, but perhaps also on the weight at the same time? Now, how can we do that? We can, for the moment, neglect that there could be a relation between diameter and weight, and we can just look at the question, is there a difference in average diameter at each site? So this is what I found. And by the way, these orchards were in the United States. I got the data from a researcher in the United States, in Pennsylvania. And so I calculated the average diameter in the Bayesian way, which means that I also have the uncertainty in that calculation. So what you see here is the posterior distribution of the mean, the average diameter, for 3 sites. So, and again, the width of these distributions indicates the uncertainty that I have about the actual value of the mean. I also learned about the standard deviation, which is not indicated here. This is the mean, this is not the whole distribution, but I also found out the standard deviation of the whole distribution. Now, if you look at these results, would you conclude that there is a difference between the sites? At least the means are different, so maybe you would say, yes, there is a difference. But how big is the difference? And then what is a useful message is that you should not just compare the averages. So I see here you have also the numerical values of the means. But if you are interested in the differences in the means, you should calculate the mean difference between the means. And how can you do that? Well, actually very simple. You can just subtract 2 posterior distribution, and that is called the contrast. So the contrast is the difference between 2 posterior distributions. If you do that, so here are again these means, and now I have subtracted side BD and side BV, which is here. And well, you can do it for every comparison. So I have three comparisons. And if you then look at the difference in means, you see that they are indeed different from zero. So zero is, in this case, a little bit in the posterior distribution, but it is also, on average, much higher. Not much, but it is higher. And the same goes for the other sides. So this way you have a quantitative insight in how big the differences are between the means. And these are the contrasts. So there you could conclude, indeed, it depends on the awkward side what the means will be and how big the differences are. But we can do more because we now know the means and the standard deviations from each for each awkward side. So we can not only calculate the mean, but we can calculate the whole distribution if we assume a normal distribution. It could also be a log normal distribution. So if you do that, then you get this result. So now I have sides RS and BV, and I calculated the whole distribution, so not just the mean, but the whole distribution of all apple samples that I have. And then I see that even though there is a mean difference that seems significant, if you look at all the apples, you see that there is quite a big overlap between the sides. So even though the mean could be higher on one side, it does not mean that every apple on that side is also higher, because actually quite a few are actually smaller. So that's what you also can do with this kind of analysis. So you get much more insight in what the differences actually are, and that is what we should look for, because you can say that there is a significant difference in means, but what does that mean? And here you see the actual results. So the conclusion is that even though the average diameter is 2 millimeters larger on side RS, there are still almost half of the apples on side BV are still larger than on side RS. So that is quite a different conclusion than when you only look at the means. And you see that distribution of that contrast you see here. And indeed, there is a lot of overlap between all the values. Now, we have only looked at weight at the moment, but actually our research question was also, is there an effect of weight on the diameter? But we now also have seen an effect of height. And that is a confounding factor. And in some sort of statistics and causal reasoning, they call this a backdoor path. Which is another word for there is a confounding factor here. Height, awkward side has an effect on weight, but also on diameter. And the effect on diameter we just have seen. You can close such a backdoor path by only looking at this relation, which means, and in the statistics they call that stratifying, which means that you do separate regression for each side, and that if you do that, then you close that so-called backdoor path. And then in that way, you can unravel the effect of height on weight and on diameter. If I do that, that's what I have done here. So I have done the regression lines for each side separately. And then you see, if you do that comparison per side, you see a small difference between the regression lines. And again, you can calculate the differences between the lines, and that is what you see here. And that's again a contrast. And then you see that the contrast is different for these three sides, which is shown here. So the sides are really different in that respect in comparing them. So once again, this is a way in which you can do contrast analysis to unravel the effects of different factors on a relation you want to investigate. So doing this kind of analysis give answers to the three questions that we have. Is there a treatment effect, in this case of sight? And the conclusion is yes, there is an effect of sight on the average diameter. And there is also an effect of sight on the relation between weight and diameter, because otherwise these regression lines, the contrast of the regression lines should be zero. Now, how large is the effect? Well, we also have seen that. It's rather small. It's in the order of a few millimeters, but there is an effect. Whether or not that is important, that was the third question, that really depends on your research question. Is 2 or 3 millimeter different? Is that significant, yes or no? That's for you to decide as a researcher. To me, it seems that it is only a very minor effect. There are no big effects of orchids' sites. But I'm not an apple researcher, so maybe someone else who is an expert values that different. But in any case, we do not let a p-value decide whether or not it is significant. We need to decide that. Okay, that was the first example. The second example is on eating rate. Lise Heuve is a PhD student here in Wageningen who is studying the effect of food hardness on the eating rate. And the reason behind that is that there is this hypothesis that foods that you need more time to chew on, you eat them slower, and therefore you also will eat less. That's the background of this research. Now, in order to investigate that, she did different treatments. She had hard carrots, she had soft carrots, and she also had hard carrots with sauce and hard carrots without sauce, and soft carrots with and without sauce. So there are quite a lot of treatments, and her response variable was the eating rate. How fast do people eat? And she could measure that in grams per minute. And that research has been published, meanwhile, in Food Quality and Preference. You see the reference here. Lise was so kind to give me her data because she presented it at one point, and she presented an ANOVA table, which looked like this. So she did this classical ANOVA that we already discussed at the beginning. And these are her results in terms of the different treatments. And her conclusion was from this ANOVA analysis, if you compare soft and hard carrots, the eating rate, ER stands for eating rate, goes up by 80%. So soft carrots are much, much faster eaten than hard carrots. This is not a strange conclusion, but that's what she also found with this ANOVA. And the p-value for that is very low, which means, again, the null hypothesis is there is no effect of soft and hard, but the p-value says, well, that is very unlikely. She also concluded that if you add sauce to the carrot, this increases the eating rate with about 30%. And again, a very low p-value, meaning that the null hypothesis that there is no effect of sauce is not very likely. So this is the standard way, which you also see a lot in papers. This is the classical way of showing treatment effects. But when I saw these data, I thought, maybe it's also interesting to do this contrast analysis. And so she gave me the data, and I could do this contrast analysis. So this is what I did, but now this is the Bayesian approach. I did this analysis with the same data. The data are exactly the same. And here you see the means again. So these are not the whole distributions. These are the means of each treatment. But then with the posterior distribution of the mean, not just a point estimate, but the whole distribution. So you can see that there is some uncertainty in this eating rate, but you also see that bit. They overlap a little bit, but not too much. So the conclusion from this would be that there is indeed a difference in eating rate between these 4 treatments. But what I just showed for the apple example can also be done here. We should not look at the means differences. We should calculate the mean difference, which leads us to contrast analysis. And that's what I did in this figure. So here are the means again, but here are the contrasts of the means. And I can then do that for comparing each side, which leads to this result. And if the contrast would be 0, it means that there is no effect. But you see that the posterior distributions of the contrasts are all above 0, which means that there is an effect, but also that there is a difference between the different treatments. And the biggest contrast is between the soft carrots with sauce and the hard carrots without sauce. Also not surprisingly, but that comes very nicely also out of this analysis. But I think the big differences here in comparison to ANOVA, you immediately see also visually how big these effects are. And whether or not this is a very significant effect is something to judge by the researcher. So a Bayesian analysis does not tell you this one is better than the other one. It's just showing you the outcome, and you will have to decide if this is valuable or not. I also did the analysis with these data by not only looking at the means, but also at the whole distribution. And then if you do that, you see that there is still quite a bit of overlap. So there are still people who show the opposite effect. And that's also, again, I think, useful information. Not everyone is having the same eating rate. It really depends also on on the person that is giving you these data in this case. And here you see that about 5 to 6% of the people actually show the opposite of what the main conclusion was. And this is definitely true if you look at the smaller differences. You see that almost 30% of people show the opposite results than you would conclude from the mean. So again, this Bayesian analysis gives you a much better insight of what your data are telling you than just a few numbers and the p-value. Something that was also interesting is that these data, I should also, let me first say, if you do ANOVA, which is basically linear regression with sums of squares, the assumption is, as we said yesterday, that the variances are all the same. And actually, that should be tested. So if you do different treatments, the variances in these groups should be the same. So I checked that for the data of Lise, and here you see the spread in the raw data. So I did not do anything with these data except for plotting the density. And then you see that there is quite a difference in the variation here. So especially the soft carrots with sauce show an enormous difference in how people have their eating rates. The hard carrots without sauce are showing the less variation. But the other ones are actually not so similar in variance. So actually ANOVA is also not the right the right analysis because the assumptions are not fulfilled. So by doing this Bayesian analysis, you get, I hope you see that, much more insight in your data and in the outcomes of the analysis. So the conclusion about this part is that a Bayesian contrast analysis is actually giving you much insight, but also quantitative insight, not only on averages, but on the whole sample. And as I also have showed, you don't need p-values or f-tests to come to a conclusion. You can just show these graphs and tell your audience, okay, I do see a difference here, And well, whether or not that is significant is up to the researcher to decide. I should say that contrast analysis is also done in the frequentist world, but then there are no distributions possible. You cannot make posterior distributions because that is not possible in the frequentist world. You only get point estimates. But I would say that whenever you are interested in seeing if there's a difference in treatment, do this contrast analysis. It's very simple, and it gives you a lot of insight. So that is my conclusion when comparing ANOVA and contrast. Once again, ANOVA is not wrong, but it has very strict conditions, and you only get point estimates and p-values. Okay, then I wanted to finalize my contribution to this webinar by telling you something about multi-level modeling. But maybe this is a good time to have a 5 minutes break. And then I will finalize with this multi-level modeling technique. Okay?
Yes, it sounds perfect. Thank you. So we
Okay,
can
see you.
reconvene at 9:40.
Yeah, okay, let's do that. Okay, I think it's time to start again.
Yes, here we are.
Okay, great. So the last part of my contribution is some small introduction to a technique that I only discovered maybe some 5 years ago, and I became very enthusiastic about this, and I think it needs much more use in the food sciences. So that's why I'm wanted to share this idea with you. So it is a relatively new modeling technique called multi-level modeling. There are other names as well. I will talk about it later. And um, the reason it's also fitting well in what I told you about the Bayesian approach is that it is very well suited for multi-level modeling, because multi-level modeling is about variation of parameters. But I need to explain to you what it is and also why we should use it. And the reason I think why we should use it is that we are, as food scientists, we are faced with a lot of variation at all kinds of levels. If you are a microbiologist, you know that the same strain, uh, even though it's the same strain, does give you different results if you do experiments. If you pick apples from the same tree, they are not the same. If you collect samples from milk from the same herd, they are not the same. And certainly not when you collect milk from different herds. Um, so there is a lot of variation here at also depending on the level on which you are measuring. Is it in a tree, apples from the same tree, or is it apples from the same orchard? That does matter. Also, for consumer sciences, it is important because consumers are, as we all know, not equal. People have different feelings, they have different taste buds, they have different sensitivity to smells. So everything that they report to you back is variable. So that's one reason why multi-level modeling becomes interesting, because it tries to tackle that variation, at least to characterize it. So here is an example that you all will recognize. I think this is I just took this from I think from Combase, where salmonella, the same strain of salmonella is studied and results are published, and then you see this enormous variation between 18 experiments. Um, Another example from food science, this is about the oxidation of ascorbic acids, where researchers in France, and this is not even in a food, this is in a model solution, where they analyzed the same solution. No, that's not what I should say. They analyzed 15 solutions of the same concentration and the same conditions 15 times, and this is the result. You see enormous differences between one experiment and the other, even though the experiment as such is completely the same. They try to do it very reproducibly, but still you see this variation. If you do something with nutrition or with pharmacology, this will also be familiar. If you do a study where you see how a food component, for instance, ends up in the blood, and you do that with people and you measure concentrations in blood, and you give exactly the same drug in exactly the same way, you still find variations. And this is an example for a drug where you see that the same drug is differently metabolized in different persons. They all show the same trend, but the variation in each individual is quite remarkable. So that is one reason why we could why multi-level modeling becomes interesting. But there is another problem that people do not always realize, which is probably also very important, that if you do measurements like the ones I just described, they are correlated. So to take this example of this vitamin C degradation, these measurement points are from a reactor. So each point here comes from the same solution. It's just followed over time. If you do the same thing with bacteria, and you would sample from the same culture solution or suspension, then they are correlated. What you measure here may depend on what you have measured here. Also, in measuring these biological samples in blood, these measurements are correlated because it is the same person that you are measuring over time. So if you measure concentrations or number of microorganisms in the same sample over time, you are probably faced with correlation between the measurements. And if you remember my lecture of yesterday, one of the conditions for regression was that the measurements and the errors should be independent, and they are not. And the same goes if you ask consumers about the food, for instance, to test the shelf life, and you use the same consumer, these measurements will be correlated. So that is a big problem that is not always realized. Then there is another phenomenon that I see sometimes in literature, and that's called pseudo-replication. And let me just take one simple example again of the apple. If you measure an apple 6 times, the one apple, for instance, to measure the weight over time, that is actually pseudo-replication. You think you might have 6 measurements, but you only have one because you use one apple. And so you really have to be very careful with this kind of experiments and be aware of this kind of problems that you might have, because the result is that your assumptions for regression are no longer correct. And therefore, your results may be biased. And that's, of course, not what we want. Okay, so what is the solution for that? Well, multi-level modeling, of course, but why is that a solution? If you think about what the examples I just gave, you can recognize groups. For instance, with the vitamin C analysis, each experiment is a group. If you do microbiological inactivation, each experiment is a group. If you look at this drug response, then each individual forms a group of measurements. Now, in the classical regression, which is single level as opposed to multi-level, there you only have one source of variation, which we express in the residual error or residual standard deviation, sigma. And we call that a fixed effect because we are assuming that everything that we measure is valid for the whole population. But now suppose that we are able to group these data. And then the level we talk about, the multi-level, the level in the word multi-level refers to groups or clusters of data. And multi-level then means, of course, that you have more than one group. And then the nice thing about multi-level modeling is that we can actually distinguish between variation within a group and between groups. And what we are basically doing is partitioning the variance that we observe. So let me just give you a first sort of hypothetical example. Suppose that we have measured something, it does not matter what it is, and we have measured it in 3 groups, and this is the outcome of the response Y versus the response X. Now, if you neglect the fact that these are 3 groups and you just pull everything together, you get this result. And if you do now regression, you'll find a relation between X and Y that gives you this result. But if you would take each group separately, you have the complete opposite result. because now you see that this relation really is different in if you consider it per group. And this effect is already known for quite a while, more than 100 years. It's called the Joule-Simpson's paradox, but it seems to be forgotten all the time. So this kind of results are very dangerous if you do not recognize that there may be groups or clusters in your data. So how to deal with that? What if you see such a result like the microbial inactivation or the vitamin C analysis? The question is what you would do. What I see frequently in literature is that people, of course, acknowledge the variation, but what they then do is they just take averages. And that's also what these people who published the vitamin C data, they just took the average of everything. And the question is, you think about for yourself, what would you do? Would you also do this kind of thing? So averaging, possibly. But another approach would be maybe you should analyze each experiment separately and then see if you can make sense out of that. Or, as we just saw in this previous 3 groups example, you can also try to pool everything together and see what happens, with the danger that you are completely missing the relationship. So what I see a lot in food science literature is that people start to average if they have this variation, and they also start to normalize initial values. So it's also not only the points in time are a variable, but also the initial values are variable. And what many people do then is they just hide that by normalizing, by dividing the number of microorganisms by the number of initial microorganisms, and then you get rid of this variation at the very beginning. But that is, of course, just hiding the fact that also these initial values are variable. And so therefore my message is, if you do this, if you average and if you normalize, you actually are throwing away information. The opposite thing of trying to pull everything together is also not a good idea, because then you ignore possible differences between the samples, between the groups. Also, doing each experiment separately is ignoring the fact that they may have something in common. If you do it separately, you ignore information from one regression to another. And so the alternative approach is what is called partial pooling, which is done in multi-level modeling. What you then do is that you do acknowledge the variation, but you also acknowledge the similarities between the experiments or observations. And basically what you are doing then is that you are that the regressions are learning from each other. They become connected, and the information from one regression is used in the other regression. And that is what is called multi-level modeling. So most of the data that we collect come in groups or clusters. And so if you can identify those, that is very helpful. And then comes the discussion that I just mentioned. How do you deal with that? Do you average? Do you analyze them separately, or do you pool everything? So these results that were published in literature, they normalized the beginning, so all the variation at the beginning was gone, and they also took the average at each time point and modeled that one. Now, fortunately, I knew the authors of this article. She followed one of my kinetics courses. So I contacted her, and she was so friendly to give me these raw data. And together we actually applied the multi-level modeling to this, which was very insightful. So normaling and averaging, as you see here, the variation at the beginning is completely gone now. These are the vitamin C data, by the way, but you could also have the same thing for microbial numbers. So they just took the averages. They did show the variation, but they just modeled the averages. And then this is the dark blue is the confidence interval, and the light blue is the prediction interval. And what you do here is that you remove information, and you also give false confidence, because these prediction intervals that you calculate are not corresponding to what you actually measure. So this is not a good idea. You can also try to pool everything, and then you still, of course, have a confidence interval for the mean that is more or less the same, but now the prediction interval is very wide. If you do that, you also neglect something, namely that all these experiments are the same. They are just different experiments. And if you neglect that, if you do not acknowledge that there may be small differences between the groups, then you are what is called underfitting. If you analyze each experiment separately, this is what is done here, you see that you can actually fit each experiment quite nicely. But what you do now is that you ignore the differences. So look at this, for instance, 13 and 15. They are completely different, but you just ignore that. And if you do that, this results in overfitting. You are putting too much trust on each experiment. So therefore, partial pooling is the alternative, where, as I just mentioned, you acknowledge that each group has different variation, but you also make it possible that regressions inform each other. Now, how do you do that? You have to have a model again. So what I took here, but it could be any model. This is just an example for this vitamin C problem, which has a model that describes the concentration over time as a function of time, t, and it has three parameters: the initial concentration, the order of the reaction, and the rate constant for the chemical reaction. So those are the three parameters that we are interested in, that we want to estimate from the data. Now, what we do in multi-level modeling is that we say, well, these parameters are allowed to vary. So we give each parameter also a probability distribution. And this is called a hyperprior, because we are putting distributions on parameters that have of their own also a distribution. So it's a distribution on a distribution. And these parameter distributions are then connected by a multivariate normal distribution. Multivariate because we have more than one variate. So I'm not going to show you the details of how to do that exactly, but there's software to take that into account. But this is the basic idea between multi-level modeling. You put hyper priors on the actual priors, and then you can learn from the results. Of course, you need to be able to distinguish levels in your data. And if you do that, then the first level is always that of the actual measurement or observation. So in our example, in each experiment, the vitamin C concentration is measured, and that is your first level. The second level is your cluster. And the cluster in this case are the different experiments, because these experiments are all the same. It leads to variable results, but they are basically the same. So that is the second level, the group or the experiment number. Well, then you could also add other levels. For instance, you could do this at different temperatures, and then you have a third level. So you can have quite a few levels if you like that. But the basic message here is that as soon as you can identify groups in your data, then multi-level modeling becomes possible. Now, this is the outcome of this analysis for these experiments, and I think that is quite insightful. I'm not showing the details of how I did this, but I show you the results. Here you see the outcome of the multi-level modeling. So you'll see that we have an overall outcome, which I call the ground mean. But then I can also plot the posterior distributions of each experiment using this multi-level modeling approach. And then you see that, well, for quite a few of them, it's actually the same in terms of initial concentration, but some are also quite different. If you look at the order of the reaction, the overall mean is very close to 1, but you also see here experiments that have a very low order, and you see experiments that have quite a high order. So this tells you that what is variable here is mainly the order of the reaction. If you look at the rate constant, there is some variation, but overall they are quite the same. So the conclusion from this is that the reason why these experiments lead so to variable results is because the order of the reaction is changing all the time. Now, why that is a different matter, but at least it gives you a quantitative idea about variation of your parameters. So what is achieved from multi-level modeling? You can have estimates for all groups, for the whole population, but also for each group separately. And the very nice thing about this is that in this approach, the measurements that are correlated, because they are probably correlated, that is automatically taken into account because of this multivariate distribution that is assumed. So this kind of measurements gives you, in that sense, no bias in estimates. And what it basically does is it balances between overfitting and underfitting. If you do complete pooling or only separate pooling, separate analysis of each experiment. You also have quantitative characterization of variability, and your uncertainty estimation becomes much more realistic. And therefore, your predictions will also be much more realistic. So, well, actually, I already told you this, so that's I can skip. I also told you this, and this is the result I took for 2 experiments. So the red line is the result from taking all the experiments together, and the blue and dotted line is from the individual fits. So you can see that the overall regression is not completely the same for each experiment. So some will be below, some will be above, but this effect is called shrinkage, and it's the result of trying to prevent under and overfitting. Everything is drawn towards the general overall mean. But if you want, you can also see it per level. So it gives you a much better impression. It corrects for correlation, and it leads therefore to more realistic and unbiased estimates, and it corrects for over and underfitting. Now, in terms of terminology, I use the term multi-level modeling, which I prefer, but there are also other terms that you can see in literature. One is mixed effect modeling, which means that fixed effects are referring to the population, and random effects are referring to each experiment, and basically the deviation from the population. Another term that you see is hierarchical modeling, because there is a hierarchy in the levels. One group may be part of another group. And that's what I try to show in this graph. You can have different types of experiments, for instance, at one temperature, where you do 3 trials, and in each trial you analyze, for instance, the inactivation of microorganisms. So these are definitely hierarchical, because this belongs to that and that belongs to that. And then you can also have hierarchy here above, etc. So you can go quite far in distinguishing levels. But it is important to take this into account when you do experimental design. Now, the software that you use for this kind of modeling goes very well together with the Bayesian approach, because in the Bayesian approach, as you may remember, parameters are considered variable, and that's exactly what we do here. We assume that the parameters can be variable. Nevertheless, they also do it in the in the frequentist approach, where they use maximum likelihood estimation. Least squares is not possible, but maximum likelihood estimation is possible. But then this violates a bit the assumption that parameters are not having a distribution. So my big preference is the Bayesian approach. Now there are several R packages with which you can do that. The one I use a lot is called BRMS, which stands for Bayesian Regression Modeling Using STAM. There's another one. This is the frequentist version. It's called Linear Mixed Effects Modeling. And here you have the term mixed effects. And you also have an equivalent for the nonlinear models, which is called nonlinear mixed effects modeling. And there are also some commercial packages like MATLAB that have this option to do it. I'd like to finish with a quote that I took from my statistical hero, Richard McElrath, who says that if you are not using multi-level modeling in your data, in your analysis, you should explain why you did not do that. And now it's the opposite. I wrote quite a few articles now with multi-level modeling, and I always have to explain why I did that. But it should actually be the other way around. If you don't do it, you have to explain why you did not do it. So I used this approach already quite a lot, and here are some examples. And of course, I'm not the only one. Also, for instance, Alberto Garro from Spain is very knowledgeable about that. And we also published together, by the way. There is now quite a lot of things to read if you are really interested in that. And I think it has many, many advantages. So I would highly recommend it. So that's what I wanted to share with you in this third part of the model. So if we now can move to the questions part, that would be nice.
Yeah. Okay. Thank you very much, Professor Van Boekel. I think that just to give Federico a bit of break, it's my turn to bring this model to an ending. And first of all, pointing out that I think that all of us share that the contents of this model have been super inspiring, and I'm sure that very helpful for gaining a better understanding of statistics. That sometimes for us is a topic that I think that is often quite challenging to be applied in our field of food microbiology. So yes, thanks for your valuable contributions. We will be a bit more precise in our work and more confident in handling variability and uncertainty, probably. So once again, thank you for your valuable time and sharing your knowledge with us. So with that, I think that we can officially conclude the third session of this model. But first of all, we will move on to the question and answer session. And I can already see some questions here. So let's begin with the first one. Is comparing the mean distributions the main differences between the ANOVA and ANOVA contrast analysis? Do we gain more information by looking into the distribution?
Yes, I think you have a much better impression about how far the means are different and whether or not they overlap. So it is a visual representation of what you. I mean, it's much more clear than when you see a number, and especially if that number is just a number without indication of its uncertainty. It gives you maybe a false idea of certainty. But when you look at the distributions, you can really see how much they overlap and whether or not you can still remain with your conclusion that there is a difference.
Okay, let's move to the second one. When comparing different predicting models across multiple food matrices, it is statistically valid to use ANOVA on model residuals or error metrics. What are the potential pitfalls?
Yeah, the difficulty with ANOVA is that it has a lot of assumptions that I explained yesterday for linear regression, because ANOVA is actually linear regression. So one of the big assumptions is that all the variances are the same, and that is in many cases not true. But people neglect that and then maybe draw false conclusions. Now, I think I'm not an expert in ANOVA, but I think that there are ways to correct for that, but you should do that. And I don't see many people doing that. They just take the software and they do an ANOVA and that's it. And I think with this Bayesian analysis, you can at least show the distributions, and then you can also visually see whether or not it makes sense. So for the beginning of the question was, if you do different experiments with different food samples, then most likely the but you have to be careful.
Okay, I think it's clear. Let's move to the third one. How should we interpret the results of ANOVA when data violate the homosteticity assumption, especially in microbial count data with high biological variability?
Yeah, that's what I just said. I think there are ways to correct for the different type of variants, and but I also would have to look up how you actually should do that. But my advice would be to yeah to try to do Bayesian contrast analysis, because then you also see whether or not the variances are the same. You can just plot the densities of your data, and then you immediately see whether or not they have the same variances or not.
Okay. So also the participants are asking for expanding a bit on how contrast analysis helps quantifying to quantify the specific differences between groups means compared to just reporting overall significance.
Yeah, I think what I showed with the apple example and also the eating rate is that you can really show how big the differences are. You really see, in the case of apples, you see the difference in diameter, which is not told to you by ANOVA. It just gives you whether or not there is difference. So you actually see the actual value of the differences. And also in the eating rate, you saw these differences between the posterior distributions, which really show you in numbers what the differences are. And once again, it puts more responsibility for you as a researcher to decide, is this important or not, this difference. You're not relying anymore on some arbitrary p-value, but you have to decide for yourself. So people find that maybe more difficult, but I think it's better science.
Okay. The following one is, should we know beforehand how our data is clustered before applying multi-level modeling?
Yeah, you should be, I did not talk about experimental design, but it is very important to think hard before you go to the lab, before you start doing experiments. So now that you know about the possibilities of having how to analyze groups, different groups in experiments, you can also optimize your experimental design so that when you are having your data, you can actually identify those levels and do the multi-level modeling. So I think you need to be more careful now about how you actually design your experiments for the analysis that you plan to do afterwards. So I think spend more time in thinking and less time in doing analysis, because it's my experience that most students, they run to the lab, they want to do experiments, they want to collect data, but they did not think about what to do with those data. And multi-level modeling forces you to think about what are the different groups, what differentiates the levels. And so it's more hard thinking before going to the lab.
Okay, I think that we have a couple of minutes more for the last one. How does partial pooling in hierarchical models affect generalization across food matrices or environmental conditions?
Yeah, especially for different food matrices, I would say multi-level modeling is very appropriate because the results that you get show you how different the food matrices are. So you will get an estimate of the standard deviation within a food matrix, but also between food matrices. So if you would put everything together, you get only one standard deviation for all the food matrices together. But if you do partial pooling, you get an idea about the variation within each matrix and between matrices. So that's already, I think, a lot of useful information that helps you to interpret the results. So multi-level modeling will probably lead to more uncertainty, but that is what we want, because if we are hiding uncertainty, we are not controlling the real world. So sometimes people are afraid that standard deviations are too high, but I would say if that's the case, try to think about why that is, and maybe it's very important information. It's not bad if experiments are variable. That is some things that some people seem to think. You should not try to make everything the same. You should acknowledge the variation.
Okay. So the last one, if you are using multi-level modeling, will you tend to look at a multi-response modeling too, if you have enough data? Sorry.
Yeah, that's actually what I did. I did not have the time to explain multi-response modeling, but indeed, you can also apply it to multi-response, which I did. I also published that a few months ago. So that is indeed possible. Yeah.
Okay, so we have finished with all the questions. So if there are no more questions, I think that we can close the session. So thank you very much for all your answers, Professor. I think that everything is more clear now. I would like to kindly remind the participants some things that is to kindly fulfill the attendance list, please. It's very important for us to be able afterwards to send all the participation certificates. So please, you have the document in the chat if you have not already filled it. Together with the certificates, you will also receive that we think that are a useful resource to revisit the content, all the contents, and keep everything together after all the full series. I would like also to take this opportunity to encourage all the people to register and participate in the upcoming models that we think that we will join very highly level as this one with the Professor Van Boekel. So I won't take more of your time. Just also a last reminder to follow us in the social media channels that you are super welcome to join us. We will stay updated with all the activities that we will follow and also opportunities that may come up. Thank you very much, all of you, once again for all your attention time and participation, and also the organization team, and on behalf of our President Ursula Barron, that as Federico said at the beginning, couldn't join us today for the closing session. And finally, we will kindly ask you to please turn on your cameras just to take a group picture to sharing our platforms, please.
Yes, so let's a minute for everyone to turn on cameras if you can. Thank you again, Professor Van Boekel. These were some amazing 3 days. I'm sure everyone really enjoyed it. It was very good. Thank you so much.
You're welcome.
Okay. So let's do a big smile, yeah. And 1, 2, 3, and yes, perfect. Thank you, everyone.
Thank you.
As Olga said, so please fill the attendance form and join us, follow us on LinkedIn and follow us on our Slack channel as well to join our community and stay tuned for our next event. So with that, we close this first module. Thank you so much.
Thank you.
Thank you very much.
And of
for
course,
all
if
of
you have
you.
any feedback, feel free to share it.
Yes, also.